Good afternoon, everyone, and welcome to today's lecture. It's a real pleasure on behalf of the protein trafficking and mitochondria interest groups to introduce today's speaker, Dr. Art Horwich, who is a true pioneer in the field of protein folding. The major focus of Art's research has been on a family of large molecular machines called chaperonins that have the remarkable ability to promote folding of proteins to the native state inside an encapsulated chamber using energy from ATP. More than 20 years ago, in what was an accidental discovery during a genetic screen in yeast, Art and co-workers discovered chaperonins and showed that they are required to provide kinetic assistance to the folding of proteins to the native state inside cells. This was a major turning point in the field because up until then, the fundamental tenet based on Anfinson's early studies on this campus held that the primary sequence of a protein contains all the information necessary to specify the native fold. And while this simple idea is true that thermodynamics determines structure, we now know that it's not the complete picture because it doesn't take into account the rugged nature of the energy landscape inside cells that provides an energetic barrier to folding. So Art trained originally as a physician. He received his undergraduate and medical degrees at Brown. He went on to train in pediatrics at the Yale School of Medicine before moving to Walter Eckhart's lab at the Salk Institute to work on T antigens, followed by a postdoc with Leon Rosenberg in human genetics at Yale, where he joined the faculty and has, over the last 2 decades, continued to study chaperonins using elegant approaches to tease apart not only the mechanism of this remarkable machine, but also the forces it exerts on polypeptide conformation and folding inside the chamber, which he will tell us about. So one defining characteristic of every one of Art's papers is this seamless exchange across genetics, biochemistry, various structural approaches, and biophysics. And it was this question-driven science that Art set the golden standard for that really excited me when I was a graduate student. And so it was a real privilege and great adventure to get to work with him and Paul Sigler on a folding active state of a bacterial chaperonin. More recently, Art has gotten interested in studying protein misfolding and the roles of chaperones in neurodegenerative diseases, in particular ALS, which he will tell us about. Throughout his career, he has received many awards, including the Gaitner International Award, the Wiley Prize in Biomedical Sciences, and most recently, the Louis Rosenstiel Award, jointly with O. Ricardo. He currently works between Yale and a satellite lab at Scripps that allows for more collaborative efforts on cutting-edge methodologies and structural approaches to studying molecular machines. And it's a real pleasure to welcome Art to the NIH. The title of his talk is Protein Folding and Misfolding in the Cell.
Well, thanks so much, Char. That's really one of the kindest introductions. I wish my mom were here to hear that. So it's really been fun to come to NIH today and see a lot of old friends and make a few new acquaintances. And actually, one of my best old friends that I got to see this morning was the director, who's a pretty busy guy, but took an hour to have breakfast. And Francis and I go way back. We were fellows here at Yale together in the genetics department. And we got to spend a lot of time with each other, seeing patients and chit-chatting about this genetics service. And I guess one of the most memorable things that we undertook together was to defeat a few local thieves in New Haven. So Francis and I, as fellows, had to park our cars in a sort of parking garage that was 2 or 3 miles from the medical center. And it wasn't a very well-protected garage. And Francis ran into the problem that some thieves had decided that he had the right car battery. And so night after night, they were stealing his battery. And I was having to take him to Sears to get a new battery every night. And so now I almost understand why he takes on these giant administrative tasks. He gets a fantastic parking space where his battery is totally safe, even on his motorcycle. So that said, I guess I should come to the talk at hand. And what I would like to do, as Charu beautifully outlined, is to present a little bit about some of the sort of historic waypoints of studying the chaperonins, but at the same time bring you really up to speed on what we've done most recently and what sort of contemporary understanding of binding, folding in the ATP cycle looks like. And then I'd like to sort of transition to what we're really beginning more and more to focus on, which has to do with what looks like a role of molecular chaperones in trying to counter a misfolded protein in the context of neurodegeneration. I mean, that's mysterious to us and somewhat deflating to think that we've been studying these machines for all this time. They're supposed to protect the cell. Why don't they do that in the context of neurodegenerative disease? So that's sort of what we've been up to. Okay, so without further ado, I also would like to pay homage to Anfinsen and to state right up front that nothing I'm going to tell you would negate any of his remarkable findings carried out on this campus in the late 50s and early 60s. And so I think this still remains probably the greatest protein folding experiment of all time, which was to take an enzyme and essentially unfold it using urea and reductant here, ribonuclease, which has 4 disulfide bonds, and then simply ask if you remove those denaturants and reductant, could the protein find its way back to a native active state? And astonishingly, it could, and it could do that actually quite efficiently if you set up the conditions in vitro correctly. And so that surely supported the idea that the primary sequence of a protein contains all the information necessary for it to reach the native state, further defined also as being likely an energetic minimum. And so that is bedrock, and I think our accidental experiment really deals more with kinetics of that process, and as Charu said, the thermodynamics is well in hand at the level of the amino acid sequence. So this is the accident that originally led us into things. I'd been working in Leon Rosenberg's lab studying a urea cycle enzyme defect involving, very interestingly, an X chromosome encoded gene for ornithine transcarbamylase, a protein known to reside in mitochondria. And at that point in history, really not all that much was known. The protein, it was known, was translated as a larger precursor on cytosolic ribosomes. It had an N-terminal basic amino acid-rich signal peptide that was sufficient to direct it to mitochondria. And there was this observation from Gottfried Schatz's lab that proteins have to be unfolded to go into mitochondria. And so, of course, at that point in history, it was presumed that they spontaneously refolded to the native state following cleavage of their signal peptides in the innermost matrix compartment. And so we were actually sort of busy studying, recognition, cleavage, those kinds of things using a Baker's yeast model where we had expressed the human OTC precursor in yeast under an inducible promoter. We knew that to make new mitochondria, to be viable, you have to make new mitochondria. And so we were essentially looking at temperature-sensitive lethal mutants and asking for ones that interfered with mitochondrial protein import and the ability to make new mitochondria. And so we'd identified a signal peptidase in our genetic collection, and we're on the way to looking at things in the membrane, when one night it really occurred to us that, aha, there could be a very interesting phenotype if, for example, proteins could enter the mitochondria but didn't spontaneously refold on the innermost matrix aspect, but rather needed some sort of machine. After all, work from Hugh Pelham and a number of others had suggested that there were these heat shock proteins in cells that had something to do with being something like molecular crowbars, preventing proteins from aggregating. But maybe they could have, we thought, constitutive actions under normal conditions. And in fact, there'd been an experiment from Sheckman and Craig suggesting that HSP70 proteins in the cytosol were necessary to allow proteins to import into mitochondria or the endoplasmic reticulum. And so the long and the short of it is we could predict what the phenotype would be, namely proteins would get into mitochondria, but they would be inactive. And so, for example, we would simply look for a mutant where this ornithine transcarbamylase would be made as a larger precursor. It would get into the matrix compartment of the organelles, but there would be no OTC enzymatic activity. So we'd see the protein, but no activity would be associated with it. And my first student, Ming Cheng, once we had that idea, pulled up such a mutant from our bank of TS lethal yeast mutants within about 48 or 72 hours. And we couldn't believe the result. It just seemed too heretical. So we spent roughly a year, roughly midway through which we received a phone call from Ulrich Hartl and Walter Neupert saying, we understand you're studying some mitochondrial import mutants. How about getting together with us on the biochemistry of that? And we were absolutely delighted to do that because we felt that we probably weren't the world's best at isolating and characterizing these organelles with respect to in vitro import assays. And so I went over there and presented a little bit about some of our various mutants, and I came to this one and they sort of shook their heads and said, oh, it looks to us like it must be this. The proteins are getting stalled in a translocation contact point. They're not reaching the matrix compartment. But if you're right, this would really be amazing. And so why don't we take your strain, make some organelles from it, and see if the protein is fully protease protected, as indicating that in this mutant they reach the matrix space but are not reaching the native state. And within 2 weeks, Ulrich called back pretty excited that, in fact, that was the case. And so we carried out a battery of studies with quite a few other proteins to establish that, in fact, there was a component that had a sort of general role in biogenesis of quite a lot of imported proteins in the yeast mitochondrial matrix. And what was really exciting was that one of the first ones we studied was an iron-sulfur protein, the risky iron-sulfur protein of the inner membrane, that goes through the matrix compartment and is proteolytically processed a couple times before it winds up in this membrane. And Ulrich could show that this protein was not undergoing correct processing. So we might find one processing event to occur, but never the second one. And the protein was in an insoluble fraction from the mitochondria. Well, this protein is a monomer. And to us, that really further supported the idea that not only do proteins come through the membranes as monomers, but they might be getting acted on by this machine at the level of being monomers. And in further studies with Joachim Osterman, it was shown that a monomeric protein that we forced into mitochondria by appending a signal peptide, DHFR, was also misfolded in the situation of this mutant. And to make a long story short, when we rescued the mutant with a yeast genomic DNA library, we came up with a coding sequence that matched to the nucleotide a coding sequence that a guy named Richard Hallberg was analyzing for a mitochondrial heat shock protein. And so we sat there on a Saturday afternoon and said, well, what's the sequence of your heat shock protein, Richard? And Ming Cheng and Richard could match the base sequence precisely. And so it became clear that this protein that we collectively dubbed heat shock protein 60 was involved most likely with folding newly imported proteins, and quite a cohort of them entering the mitochondrial matrix compartment. In addition, we could show shortly thereafter that for HSP60 itself to be made, newly imported subunits had to be proteolytically processed and acted on by pre-existent HSP60 in order to make new complexes. Okay, so in the first instance, I think we never had any illusions that we were studying something thermodynamic. I think we always thought that this would be something kinetic. And the idea is that This is the sort of experiment that Anfinsen did with ribonuclease, where this is a protein that doesn't get into any kinetic difficulty. It finds its way down what's a relatively smooth energy landscape. And this is pretty describable with a lot of standard equations. Work of Alan Furst and many other investigators are able to describe the kinetics and transition states to these kinds of folding reactions quite efficiently. Rather, we felt that it was likely that the action was on proteins that have trouble to fold under in vivo conditions of things like 37 degrees and very high local solute concentrations. And so proteins could get into kinetic traps behind these minima, such that on any kind of biological time scale, they couldn't reach the native state. They'd really be stuck behind these minima. And the idea for these machines would be that they would pull proteins out of minima or prevent them from getting into them in the first place, probably by recognizing some sort of hydrophobic surfaces that would potentially be exposed, specifically in non-native states. And so before telling you a little bit about the mechanistic details, I'd just point out that there was, as we realized very early on, a broad array of these kinds of ring-shaped folding machines, the so-called chaperonin family, as John Ellis dubbed them, distinct from other classes of heat shock proteins like HSP70s and so forth that bind relatively extended states of polypeptides. These machines bind non-native polypeptides in collapsed globular states inside of the cavities of their rings. So here's the original component. We analyzed and we right away saw that it was more than 60% identical to a bacterial component implicated in lambda phage assembly, but now we know that this must be folding of particular lambda head proteins, for example. And another component that had been implicated in assembly of rubisco as an L8S8 hexadecamer in plants, simpler oligomers in other species. The work of John Ellis suggesting that this binding protein would interact with newly made large subunits of rubisco inside the chloroplast stroma and hold on to them until the protein was fully assembled as the hexadecameric form. But I think the experiments with Ulrich suggested that as opposed to, for example, an assembly function here, as had been suggested, it was rather a monomeric polypeptide chain folding function that was involved. So shortly after those experiments, Jonathan Trent came to my lab and provided a sort of linking experiment where he looked at a thermophilic protein that was actually a heat shock protein even within a thermophilic strain, and showed that it also was a double-ring chaperonin that could hydrolyze ATP, could bind non-native proteins, and could confer thermotolerance to archibacterial cells. And lo and behold, when we looked at the structure of the subunit from this component, it raised a high degree of relatedness to a component that had been known to be present in a complex in the eukaryotic cytosol, but it never had a function ascribed to it. And this is now a component that we know as TCP1 complex, CCT, TRIC. It goes by a variety of handles, but it's the eukaryotic cytosolic chaperonin that's essential for folding such components as actin, tubulin, beta propeller proteins, and others. And so there's quite a range of these machines, but I'll focus on the mechanistic workings of the bacterial chaperonin system. I should say that this system has detachable lids that encapsulate the folding chamber here. So proteins are bound in an open ring and ultimately encapsulated by a binding of a lid. These components have a built-in lid structure that functions something like an iris, and so ATP binding closes that iris and produces a folding chamber by that mechanism. So the first reconstitution of a chaperonin reaction in a test tube was really carried out by Golubinov and Lorimer at DuPont and their colleagues. And what they could observe is that if they took a dimeric form of rubisco, unfolded in denaturant, and incubated it with GRO-EL, it formed a stoichiometric binary complex. In the absence of GRO-EL, this protein crashed out of solution and was completely inactive. Wholesale aggregation ensued. But this binary complex, which had no rubisco activity, was latent for production of the native state of rubisco, such that when these investigators added GRO-ES, a single ring component composed of 10 kilodalton subunits, 7 of them radially arranged, it could bind to the tetradecamer here of 60 kD subunits, and voila, in the presence of hydrolyzable ATP, over a period of some minutes, native rubisco was produced. Quite a remarkable reaction, and we've all spent quite a few years sorting out what goes on here, and I would like to just summarize a little bit concerning that. This is just as important, however, the ability to bind non-native proteins and to some extent rescue them from becoming kinetically trapped is sort of an additional facet of the chaperonin reaction. So none of our work would have been possible without this gentleman, Paul Sigler, that Charu referred to. And we had a wonderful 10-year collaboration with Paul before his untimely passing. Here he is shown just as he was coming to Yale, looking at a trip repressor operator structure that was solved with these two wonderful guys, Andrzej Ohimiak and Zbyszek Otwanowski, who figured critically in the GRO-EL project. And basically what happened was that I got sent to talk to Paul about a university-related matter. I'd never met him. This was roughly 1990. He'd just come from Chicago. And I think we talked about that matter for about 20 seconds before he said, I saw that interesting molecule that you seem to be demonstrating can mediate protein folding. Look at all the symmetries of that thing. Have you ever thought about trying to generate crystals of that? And so within 5 minutes, the collaboration was born. Zbyszek felt that, at that point, computing could never handle a molecule of this size, but that it would probably be a real troublesome job to generate decent crystals, and by the time we were able to do that, he'd be able to handle the computing. And that turned out to be exactly right. It took us 3 years to generate a good crystal of GRO-EL. We tried quite a lot of things because we were aware that other people had played with it. We tried monomeric versions of it, trimmed versions of it, and finally, in desperation, we turned to the fully intact molecule, just substituting particular amino acids that were critical ones right down the primary structure. But remarkably, in the end, the molecule that really behaved the best, really diffracted nicely, was a molecule that we'd considered to be just a wild-type variant. It had a PCR error in it, and it produced beautiful crystals that were solved in fairly short order by Zbyszek, in fact, once we supplied him with an ethyl mercury derivative. So without belabor, and I should just say that refining this structure was a headache of a further year's duration, helped by Axel Brunger and his team. But I should say that in the end, we saw something similar to what electron microscopists had reported, namely 2 back-to-back rings with 2 major domains of each subunit in the ring. Each ring is composed of 7 radially arranged subunits. And the primary, or the domain structure of the individual subunits is such that you have these equatorial domains that essentially hold the machine together at its waistline. They make contacts going around a ring, but also across the ring-ring interface. And so this is sort of the base of the machine. Then there's a sort of smaller hinge, so-called intermediate domain that's covalently connected both to the equatorial and to these apical domains. And these domains are actually pretty much flying in the breeze. They make an electrostatic contact or two between them, but they're basically only weakly attached to each other. And so basically the machine opens and closes as a hinge when ATP binds on the inside of these equatorial domains. Looking down the barrel of the machine, you see this 45 angstrom diameter hole, but it's actually blocked off by disordered C-termini that dangle into the central cavity at the level of these equatorial domains. So it's really 2 separate chambers to an open GRO-EL ring. And they function during the reaction cycle out of phase with respect to each other. So this was the polypeptide binding surface that we identified inside of a ring. So if you were standing in the top ring of GRO-EL, as I just illustrated it, and looked at the back wall, all of these hydrophobic residues are facing solvent. And mutation of any one of them, 7 around a ring, abolishes polypeptide binding. And so that was consistent with our sort of very early model that presumably hydrophobic contacts between exposed surfaces in the substrate protein and this proffered binding surface in the ring of the chaperonin are involved with polypeptide binding. Once a protein reaches the native state and isn't exposing any significant amount of hydrophobic surface, there would be no affinity of GRO-EL for it. It might drift into this chamber, but it would drift right back out. And so binding is multivalent in character, and we could show that in a variety of ways. First was a mutational study where we made a GRO-EL ring as a single continuous coding sequence, 7 subunits strung together. Now you could make mutations in the peptide binding site in any combination or permutation. These are actually double rings when expressed in E. coli, but what we could show is that you need 3 or 4 consecutive good binding sites that are intact and non-mutant in order to efficiently bind a non-native polypeptide. Then more recently in cryo-EM studies with Helen Sable's group, we observed that malate dehydrogenase in the non-native state binds to 3 or 4 subunits. Here it is in contact with the apical domains bound in an open ring. Other substrates have been looked at in a similar fashion, and so it seems like binding is multivalent. Now, I should point out that this is only a fraction of the total density of MDH. And this represents probably many different conformational states. Bound proteins are pretty much pulled apart, and I won't show you this, but in NMR studies, direct NMR studies with Kurt Vutrich's group, what we've been able to observe is that isotopically labeled proteins, while they're bound to GRO-EL, exhibit no appreciable secondary or tertiary structure. So the machine really melts out the non-native protein on its hydrophobic binding surfaces. And in that way, it could, to some degree, have ability to pull apart a non-native state and prevent it from getting kinetically trapped. And in fact, here's a sort of direct demonstration of such behavior. This is an early experiment carried out by the Lorimer group, where they took Rubisco and unfolded it in acid, then diluted it into a neutral buffer and asked whether it could fold in the absence or presence of the chaperonin system. And the point is that if GRO-EL, GRO-ES, ATP, the full Monty is there at time 0, you get quite efficient recovery. If you add it only later, you get less recovery and progressively less because a lot of the Rubisco has now aggregated and is not accessible to the chaperonin system. And in fact, if you never add GRO-EL, none of the Rubisco ever makes it to the native state. So that suggests that Rubisco becomes, as a monomer, and these are experiments, I should say that Hayes Rai, one of my former postdocs, has really eloquently sort of supported with FRET types of studies that I won't detail. The monomer of Rubisco, free in solution, tends to aggregate, but even this monomer is incapable of reaching the native state. It's in a kinetically trapped state, and unless it gets bound by GRO-EL and to some extent pulled apart and given a chance to fold in the folding chamber, it won't reach the native state. And so that suggests that indeed a key action of chaperonins is exerted by pulling these kinds of misfolded states apart and then giving them another chance to fold in a folding chamber. And in fact, Hayes demonstrated that very directly with FRET studies that he could carry out at relatively lower temperature than normal, but he could show that the polypeptide was pulled apart upon binding to GRO-EL on a very rapid time scale, corresponding to physiologic rates of binding. And so I think that's the best evidence concerning how GRO-EL rescues polypeptides from kinetic traps. This comes at the level of polypeptide binding. But I want to qualify that from the standpoint that the intermediates that are bound by the chaperonin are poorly structured. If you build any degree of hydrogen bonding, strong hydrogen bonding secondary structure into an intermediate, the chaperonin really can't pull it apart. So it's acting on weakly structured states. And so I think my final point along these lines is that misfolded states can have wrong topologies that are relatively stable, even though to hydrogen exchange, they're quite exchangeable and loosely structured. Okay, so now I want to turn to the folding side of the reaction. So I've mentioned Helen once before, but I want to again emphasize how critical she's been to these experiments. And the idea really brought forth by this golf course in Melbourne is that she looks at single particles in all sorts of different orientations and deconvolutes them into an image reconstruction. And that's been critical to looking at the chaperonin system. And so with respect to polypeptide folding, when we initially superposed one of our favorite substrate proteins against GRO-EL, there was a very bad steric clash, and we didn't really see how it would be possible to have a folding reaction inside of this machine. But then shortly thereafter, through reconstructive work, Helen showed that when GRO-ES binds to GRO-EL, the apical domains on that side of the chaperonin machine open up and provide a folding chamber, as it were. Here's the opposite side, not bound by GRO-ES. It's relatively closed. And so this raised the question that maybe a polypeptide could be encapsulated in there. And Jonathan Weissman, who was a postdoc in my lab at the time, quickly carried out some protease protection experiments that clearly indicated that polypeptides in this space could be protected from protease. Polypeptides bound here were completely digested, and that supported the idea that proteins could be bound in this so-called cis ring, so make a cis ternary complex, as it were. And we could discharge cis and trans ternary complexes. Here, GRO-ES is opposite to the bound substrate protein, therefore a trans complex. We could discharge those complexes with ATP and ask which was productive under single turnover conditions that I won't detail. The bottom line was that this complex is productive and this one isn't. So polypeptide folding, at a minimum, had to start from this kind of cis ternary complex. Moreover, I should say that in a single ring experiment with a version of GRO-EL where we've basically mutated the interface so that we have just a single ring version with GRO-ES bound, we could watch in this stable ternary complex a substrate protein go all the way to the native state. Inside the complex, we could gel filter it and see activity at the position of the 400 kD complex. This was an experiment carried out by an undergrad, Corinne Hole, who was rotating through my lab. But the point is that the single ring version of GRO-EL is very stable because the normal ejection notice for GRO-ES discharge is binding of ATP to the so-called trans ring. Well, if there's no trans ring, this so-called cis ternary complex is a stable folding chamber. And Corinne could show that proteins reach the native state in that chamber, which is stable on a long time scale. So what does that look like? This was the work of Xiaowei Zhu, a postdoc of Paul's, who worked out how to freeze these asymmetric complexes. This is GRO-ES bound to a ADP-bound GRO-EL complex. And what Xiaowei observed was that the trans ring looks just like an unliganded GRO-EL ring. The cis ring undergoes these large rigid body movements where the apical domains are elevated to contact GRO-ES. Here you see in a top-down view that GRO-ES has its own little loops that have hydrophobic sequences in them that now make contact with that original hydrophobic binding surface in GRO-EL that's moved through space to proffer itself to interact with GRO-ES's mobile loops. And so this is all rigid body movements of a large order that are summarized here. Roughly a 60-degree elevation of the apical domains and a 90-degree clockwise twist induced by nucleotide binding, such that the initial hydrophobic binding surface facing a cavity that would be here is now facing us out of the board, and there's a completely new binding surface here facing the cavity. And the nature of that surface is basically a hydrophilic one. This just shows first, that you've gone from a 45 angstrom diameter open hole, and I should say that bound proteins can drip out over that hole like a champagne cork, shown by neutron scattering studies. But once encapsulated, this cavity is enlarged to be about 60 by 60 angstroms, able to house polypeptides up to maybe 40 or 45, 50 kD in size. and the protein folds basically in isolation here. There's nothing for it to aggregate with because the walls are hydrophilic and we believe probably don't interact very significantly with the non-native polypeptide. It's going to translationally bump into that wall, but it isn't likely to physically bind to it. And so the protein simply folds in effect as if at infinite dilution with nothing to aggregate with. So here again is a look at that same structure. All this blue represents hydrophilic surface. It's a net negative charge, but gigantic numbers of both positive and negative charge side chains. Here's the trans ring where polypeptide binding would occur initially. All this yellow is hydrophobic surface that recruits non-native proteins. And so the machine is typically in an asymmetric state, and that's a function of cooperative binding of ATP within a ring with a Hill coefficient of roughly 3.5, and strongly anti-cooperative binding of ATP between rings. So when this ring is loaded with ATP, which is required to recruit GRO-ES, this ring will be not able to be occupied with ATP, will not bind GRO-ES, and experimentally will not bind non-native polypeptide until ATP in this ring is hydrolyzed. So this is just a comment on what the minimal folding active state of the chaperonin is, namely an ATP-bound state that moves the apical domains, allows GRO-ES to bind, and ejects polypeptide off the cavity walls into the folding chamber. This is a folding active machine, experiment carried out by Hayes while he was working in the lab. And one of Charu's major contributions was to show that the gamma phosphate of the ATP is absolutely crucial. So she could nucleate a machine that looks like this in ADP and GRO-ES. The polypeptide stays stuck on the cavity wall. ADP is insufficient to produce a sort of forceful apical movement that would eject polypeptide off the cavity wall. But if you add either this transition state metal complex or even a ground state metal complex, of beryllium fluoride, that's enough to trigger a movement that ejects polypeptide into the chamber, and now it folds the same as it would in a normal folding reaction. So this effectively simulates the addition of the gamma phosphate as a separate step, and really, I think, elegantly shows that those 7 or 8 hydrogen bonds formed by that metal complex produce enough energy to eject the substrate into the folding chamber. So this is a look at the folding chamber, and here you see all these electrostatic residues. It's pretty much devoid of hydrophobics. We've mutated these. There's the 2 or 3 that are left, and there's no effect on folding. So a major question in our field has been whether this charged cavity is actively assisting folding or behaving passively. So it's been noticed that some substrate proteins can fold up to 10 times faster with the chaperonin system than free in solution. Is this due to an active assistance by this machine? And I want to show you a couple experiments that make me feel that that is not the case. So here's a look at a double mutant of maltose binding protein folding under what are called permissive conditions, where the protein can either fold free in solution to the native state or use the chaperonin system to reach the native state. And so what you see is that it's quite slow free in solution. This would suggest that the chaperonin might be capable of accelerating the reaction. But that's not the case. What is the case is that the protein is really decelerated free in solution because it's reversibly aggregating. So here's a look at wild-type MBP. No concentration dependence to the rate constant of folding free in solution. Here's the mutant MBP. Concentration dependence of folding free in solution. Telltale sign of aggregation free in solution. As concentration goes up, the rate constant goes down. Dynamic light scattering shows wild-type MBP, DMMBP, no aggregation. Wild-type MBP, the double mutant, light scattering immediately upon dilution from denaturant. It's subject to aggregation directly observable. So Adriana Petri in my lab, fooling around with the protein folding free in solution, noticed that if you omit chloride ions from the refolding buffer, the aggregation goes away, no light scattering. What is the rate constant for folding under those conditions? It accelerates to exactly the rate that's observed in the chaperonin system. To me, this indicates that we're observing the following. Free in solution, there's reversible aggregation going on. this off-pathway step slows the folding of DMMBP substantially, tenfold, to reaching the native state. But if you cut this off basically by either omitting chloride ions or by using the chaperonin reaction where you're folding in solitary confinement, now you observe only this intrinsic rate of folding to the native state that I just described. So this is an overall look at the reaction cycle as we know it now, incorporating a number of things that we've published during the last 6 months or so. So the first thing that we've observed fairly recently is that the acceptor state during the reaction cycle is probably not this asymmetric ADP complex with an open ring. This is relatively less efficient at binding non-native polypeptide. It's rather a rapidly formed ATP-bound complex where ATP has entered this ring, caused the apical domains to move a little bit, and now polypeptide binds to these mobilized apical domains far more efficiently than it does to the non-mobilized ones. That's not to say that this can't be forced to become binding active, but under physiological conditions, this is probably the acceptor state for non-native protein. Then GRO-ES is recruited in a next step, forming some sort of a collisional state where polypeptide and GRO-ES are both in contact with the apical domains. It's very short-lived. We're talking about something that's probably occurring 200 milliseconds into the reaction and has a lifetime of only another 100 milliseconds. And then there's a very large rigid body change that is described by the structural features I showed you that ejects polypeptide into this cavity where it begins to fold. And that's done by 1 second. This is a slow sort of conformational change that takes a good chunk of a second, but you're moving a huge number of cubic angstroms of polypeptide. Then comes the longest step of the reaction cycle, really the duty cycle here, where polypeptide is folding in the cis cavity, I believe, free from the cavity walls, basically, unperturbed, more or less at infinite dilution. And at the end of that time, whether the protein is folded or not, hydrolysis occurs, gates entry of ATP into the opposite ring, and that ejects all the ligands from the cis ring, and that's the end of the folding cycle. So polypeptide goes into solution. If it's native, that's it. It finds its partners and works in the cell. If it's non-native, it can come back to the open ring or to another chaperone or to proteolytic degradation machinery. So I want to just show you a quick sort of movie that connects the dots, as it were, of all the crystal structures and the cryo-EM structures and morphs between them. This movie was made by George Farr, one of the senior guys in my lab. So here you're looking down the barrel of a GRO-EL here, and you see the hydrophobic binding surface at the level of the apical domains. Remember, things are blocked off at the equatorial level. And now you're going to see a ripped apart, computationally ripped apart DHFR come in. This is ATP binding, which is going to move the apicals to this favorable position where the hydrophobic surfaces are mobilized to efficiently bind DHFR. And this will be followed by GRO-ES binding through its own hydrophobic surfaces, which triggers this huge conformational movement. It's a 60-degree elevation, 120-degree clockwise twist. That ejects the polypeptide, which begins to fold a la Anfinson's rules. And now this is the longest step of the reaction cycle, at the end of which hydrolysis occurs. Out comes the free phosphates that gates ATP entry into the opposite ring and starts to move the apical domains here. Now you get efficient polypeptide binding on this side. But at the same time, this ATP binding has sent an allosteric signal that ejects the ligands from here. Probably the order is GRO-ES polypeptide and then ADP moiety. And meanwhile, this ring is folding active. So I just want to show you this in what we think is more like real time. And you'll see that the machine spends most of its time in the folding active state, like this. And then essentially, the transition from this ring being folding active to this being folding active occurs in about 1 second. So ligands come off and on come the other ligands, and now this ring is going to be folding active. And so we think that's the basic workings of the machine. Okay, so I want to spend my remaining minutes to talk about some newer work that we're doing related to protein misfolding in motor neurons. So while we've been busy trying to understand mechanism of chaperonins, a whole industrious group of people from neurobiology has been essentially learning that many of the common neurodegenerative diseases are associated with protein misfolding and characteristic aggregation. And I won't enumerate here, but it's shocking to us to see that a number of these aggregates are occurring in the cytosol, where there should be a good supply of all of our favorite chaperone family members. And I was particularly horrified to learn that SOD1, a small homodimeric free radical superoxide scavenging enzyme, was associated with ALS. And this was really brought home to me when the guy that teaches my little kid tennis started to limp around the court and died over a period of 1 year. And I kept reading the literature and kept coming back to this. And then J.O. Wong came to my group as a postdoc from David Borchelt's group at Johns Hopkins and started to tell us about the work he'd been doing and what did we think he should do. And we said, well, we're pretty interested in what you were doing, J.O. Why don't we all shift to what you're doing instead of you shifting to what we're doing? And that's pretty much what has happened. So to reiterate, in ALS, be it SOD1 induced or sporadic, there's cytosolic aggregation observed. And sporadic cases have increasingly been strongly associated with aggregation of this protein, TDP43, in the cytosol. This was originally work from the Trojanowski and Lee group in Philadelphia, and I won't dwell on that. I think it's a really interesting observation. The question is TDP43 aggregating in some SOD1-like fashion, or is it doing something completely independent? And I won't dwell on that question. But I just want to say that both in that situation and the SOD situation, you see co-staining of aggregates with ubiquitin. And that's pretty universal in all of these aggregation-based neurodegenerative conditions. So SOD1, as I mentioned, is a homodimeric enzyme. Here you're looking at its active site. So what it does is it can handle 2 molecules of superoxide for each sort of traverse of the enzyme. So it can oxidize one to an oxygen and then reduce a second one to H2O2 by essentially using this copper. So the copper first gets reduced and then reoxidized, if I'm getting it right. The protein also has a disulfide bond, which is interesting and very handy for reporting on whether the monomer is folded or not. It's a misfolded subunit if you don't see this disulfide. And I should say that the disulfide is there despite this being a cytosolic enzyme. So one of the first things we did was to make fusions of wild type and mutant forms of SOD1 and fuse YFP to the C terminus so that we would have some sort of optical reporter. And actually we did these first experiments in worms, and I'm just going to say 1 or 2 things about that because of time constraints. But when our worms developed a phenotype, we then said, oh my gosh, we better make mice with these fusions and make sure that they really do get a phenotype. So we used human genomic clones to do this and made transgenics that are basically a wild type and a mutant. And I should say that G85R is a selected choice because Olofberg, Markland, and others in Sweden have shown very clearly that G85R is a completely misfolded monomeric polypeptide. It never makes the SOD dimer, it never gets metallated, and it has no enzymatic activity in our hands. So we think it represents just a badly misfolded protein. It is notable that SOD, as you saw a slide ago, is this beta sheet rich affair that looks like a TTR or some sort of an amyloidogenic protein, but I don't think that we think of SOD-linked ALS as a real amyloidopathy, and I don't think that the aggregates are true amyloids. So when we looked at our mice, this is one of the early animals, sure enough, they are, by 8 months to 9 months of age, they have lower extremity paralysis, and this mouse would really only live a few more days without major support. So then we started to look optically to see what was happening. And what we saw was a progression. If you took a cross-section of spinal cord and looked at the gray matter, you saw these large neurons, typically in the anterior horns, with pretty strong fluorescent staining, but diffuse in the cell bodies and a fair amount of staining in processes as well. And that was pretty much what we saw at 5 or 6 months of age. but at 9 months of age, we saw catastrophe. So this is a symptomatic animal reaching the bitter end. You see aggregates all over the place. So inside the cell body, there are punctate aggregates in the cytosol. There's a lot of aggregated material outside here in the neuropil and the gray matter, and there's even a little bit out in the white matter zones as well. So aggregation has really supervened very strongly at the tail end in these animals. This is to just show you that we observe the same kind of effects in worms. So here is a mutant worm, and here's a wild-type SOD-YFP worm. So this is moving the same way an N2 Bristol wild-type worm would move. And this guy just basically is a standard G85R worm. It just rarely makes a movement, and not much of one at that. And of course, we're using the power of genetics to further analyze these guys, and have isolated a rare suppressor now that we're characterizing. So this guy turns out to be a double mutant. It affects a protein quality control mutant, but also a gene expression gene. It's a little too soon to talk about it, but that basically allows the worm to reverse the toxicity of the misfolded protein. So it still misfolds the G85R protein. That is not a fusion. That's just G85R or wild-type SOD without the YFP. It still aggregates the G85R, but it no longer has toxicity and it moves as a normal worm. So we're going to be interested to try to understand that. So I want to spend the last few minutes just talking about mouse observations. So in the mouse spinal cord, what we saw consistent with the optical reporting was progressive insolubility at a biochemical level. So here I should say that the major expression that we observe for this particular fusion protein is in motor neurons. So when we do in situ hybridization, virtually all of the RNA signals we see are coming from large neurons and not from glial cells. And so when we're looking at a biochemical level, we're really looking at what we believe is expression mostly in motor neurons. So here's a wild-type transgenic animal, same construct, the SOD-YFP fusion. Everything is soluble. And here's the mutant at 1 month. Everything is soluble. I should point out that G85R fusion, or even non-fused, runs a little bit faster than wild-type and SDS gels. At 6 months of age, we start to see insoluble material. And at 9 months of age, there's a huge amount of insoluble material. And I didn't show you this slide, but I want to say that if we do gel filtration on these spinal cords, by 6 months of age, we start to see soluble oligomers of anywhere from 100 kD to 800 kD size, just a range of soluble oligomers. None at 1 month of age, and strong once again at 9 months of age. So animals by 6 months of age are just aggregating SOD-YFP, making soluble oligomers and insoluble aggregates. Okay, so what we'd really like to know is where are the chaperones? So what we wanted to do was to take soluble misfolded SOD fusion and ask whether it associates with anything else. So we take spinal cord, homogenize it, do a clearing spin, take the supernatant, and apply it to an anti-YFP affinity matrix, wash, elute, and now do mud pit on that. So this is experiments initially done with John Yates, but now we have an orbit trap in our lab and are doing mass spec ourselves. So we basically take this eluted material, treat it with trypsin, and then do essentially a sequential chromatography that goes straight. It's right at the front end of the mass spec, and it goes directly into the nano spray, basically. And so this is a sensitive technique that allows us to interrogate what's stuck to the SOD-YFP in the soluble fraction. And sure as shooting, we see HSC70 associated with this misfolded protein in pretty good spectral count amounts. So here's the misfolded protein, 3,000 counts. Here's HSC70, at least 600, not changing especially with age, particularly until the bitter end. And in the wild type, equivalent amounts of SOD-YFP, but little or no HSC70 associating with that protein. And so that's the most abundant thing that we see associating with the SOD-YFP. So indeed, there is a chaperone binding. Why is that not protective? That is troubling to us, and we really don't understand that. Here's just a further demonstration that that is a specific association with the mutant. So here's an immunoblot of the eluted material from that anti-YFP column. The mutant is appreciable with an anti-HSC70 antibody that indicates that it bound to the G85RYFP misfolded protein, and no HSC70 in the case of the wild-type complex. If you use like a quarter of a spinal cord, so now you're looking at just Kumasi staining of the gel from the affinity capture, you see that HSC70 is indeed the major species associated with this misfolded protein migrating a little faster than wild type here. And this is in the absence of any cross-linking. So think about all the washing and manipulations that we do. Potentially, this is normally a stoichiometric interaction, although that's strictly speculation. But here's what we think we're looking at this point. I just don't have time to show additional data, but the misfolded protein is a soluble monomer. And at all ages in our animals, it seems to be associated with HSC70. And I don't know if I have this slide, but I want to say that we've done laser capture microdissection on spinal cord motor neurons and now subjected them total protein to mud pit analysis. And the most abundant protein in the motor neurons is HSC70. Next down the list is HSP90, a little less abundant, maybe half as much material. But HSC70 is the primary chaperone that's the defense network. And there's no induction of it over time when we look at our mutant animals. So the animals have ongoing aggregation and disaster going on, and there's no heat shock response at all. There's no HSP70 induction. The levels of HSC70 don't change. And so I don't understand that. Why are neurons not capable of mounting this kind of response? Maybe it's considered to be damaging in terms of development, or we don't understand. In any case, this mutant protein is able to be turned over quite quickly by the ubiquitin proteasome system. I don't have time to show that data, but we've done animal experiments, hydrogen deuterium exchange in intact animals, that shows that it's rapid. And it's rapid even at late times when the animals are already symptomatic. Interestingly, at the point of 6 months, when the animals have soluble oligomers in their motor neurons, they also recruit HSP110. So there are 3 exchange factors that exchange ADP for ATP in HSC70. And these are the HSP110 components. They may also be capable of directly binding non-native polypeptides. There's some evidence in the literature to support that. So we don't understand as yet whether this is actually helpful to the animals or really harmful. And we're trying to do some in vitro reconstitution experiments to look at this, as well as doing kind of transgenesis kinds of experiments in our worms. And at late times, we've carried out mud pit on insoluble aggregates, and they're rich in neurofilament chains and vimentin, but I don't think we've seen anything there that is a clear indicator of some new understanding of the pathologic process. So this was just a slide summarizing concerning total chaperone content within motor neurons identified by mud pit. Again, the most abundant protein is HSC70, and it's not very responsive to either this mutant protein, same levels in wild-type animals. There's a little bit more of it than there is of the mutant protein, but not all that much. And so the questions that I think we're left with at this point as major ones, and there are many others, I should say, is HSC70 overwhelmed such that it really can't perform its normal cellular functions as a sort of housekeeper that helps with translocation and vesicle trafficking and vesicle recycling and many other things? And/or is it just there's so much of the toxic protein that it itself is exerting toxicity within the motor neurons? And I don't think we can distinguish that with the data that we have at hand currently. Is a HSP10 helping or hindering things? Where is toxicity exerted? Is it the cell body, or is it, as many believe, that these are the longest neurons in the body, the longest axonal processes? It would be obvious that translocation and transport down those processes should be affected. We're trying to measure that now. I should say, though, that we don't see aggregates out in the axonal processes. It's like they're sacrosanct of aggregation for some reason. And finally, we've looked at the neuromuscular junction. We clearly see abnormalities at times of 5 or 6 months. where the presynaptic side of the neuromuscular junction seems to have lost its ability to stain for presynaptic markers with Bungro toxin staining postsynaptically. And that is what we see in our worms, that they're presynaptic abnormalities. So does that suggest that rather the condition is a dying back from these defective neuromuscular junctions? I don't think we know. So we're trying to untangle this a little bit more. Of course, I mentioned why is there no real classical heat shock response? What would happen if we forced one to occur? And so we're trying to do that. And finally, can we emulate the aging time or whatever that is by making ES cells and then motor neurons from them and actually see a phenotype in culture? I think we've done enough profiling now and enough looking at aggregates that if we were really seeing an ALS-like phenotype from motor neurons in culture derived from these ES cells or IP cells, if we have to do that, I think we would know it. So we can play with that, and we're interested to do that. So finally, let me just acknowledge the people who have contributed to all this work. So George and Wayne and Christina have been with me as senior people almost from day one. So I think in aggregate, we have like 60 or 70 person years together. Current people in the lab, Jao just took a job in Johns Hopkins, so is right up the street. But he was really the guy that slurped us into this more or less increasingly over months and months. First the worms, then mice, and now we're really totally locked in on this. Navneet is doing reconstitution experiments with HSP110 and HSC70. Fernando worked on amyloid aspects of things. Maria, I should say, is really contributing strongly to looking at peripheral nerve effects of the misfolded protein. And Ermi is doing both RNA and protein profiling of motor neurons. So I'll stop there, and I thank you again for having me. It's really been a wonderful day. Thank you. I could take a question or two if anybody has one.
You sort of alluded to it. Is there something, or are you thinking about what about SOD makes it the protein that forms the aggregate? Is it so abundant, or are there special properties?
Yes, I apologize for not mentioning that it's quite abundant. So it is sitting there at a level of maybe 1 to 2% of soluble protein in neurons. And in particular in these motor neurons, it's at least that. So I think that's part of what disposes for big trouble. The other is probably this beta sheet structure is another disposition. So, I mean, the question has been raised, well, why don't you just express beta globin in motor neurons and ask whether it could do the same thing? And that might be an interesting experiment to conduct, but I think we'd really like to just understand one of the physiologically toxic proteins. But yeah, that raises something interesting about the sporadic cases, because in our hands, at least in the mass spectrometer, TDP43 is not nearly as abundant a protein as SOD1. But Virginia Lee tells me that she thinks that TDP is equivalently abundant, so maybe that explains why it's being slurped into aggregates in sporadic cases. But we're trying to look at that. But I think abundance is definitely a contributor to part of these problems.
You mentioned that in the beginning that hydrogen bonding, preventing hydrogen bonding was part of your thinking about this. Does that imply that things are then in the beginning attached with more water and that there's a kind of a dehydration that takes place during this folding process?
Yeah, I mean, I suppose it's, I mean, there's just no experiments that deal with water in the chaperonin system, since the resolutions are typically 2.5 to 3 angstroms, and we've just been at a loss for how to really look at water, either during the polypeptide binding process or the folding process. I mean, there are some really nice tryptophan probes that Zuail and other people have worked out, but we just, I mean, even discussing this with him, he said, I think you're just going to be, looking at potentially a few ordered waters, and you're not going to get the big picture. So I think we could speculate. I don't think we really know. But concerning hydrogen bonding, though, I do want to emphasize that the intermediates, before they get to an open GRO-EL ring, are really poorly structured. They really don't have any stable secondary structure. I mean, their protection factors are 100 or less. And then when a protein is bound, I think it's just basically, particularly on a stable time scale, which is not necessarily what goes on in a physiologic reaction where there's just a second or so before encapsulation occurs. But in the stable experiments, the proteins are just completely ripped apart structurally and draped over the cavity wall probably. I mean, it's dynamic probably, but there's no protection visible for practical purposes. And so when we've programmed things to have some secondary structure, GRO-EL can't rip it apart. It can do it, but it takes it forever, like 20 minutes or something like that. So I don't think that it's a real unfoldase the way, for example, many of my good friends here are studying them. I mean, the AAAs have gigantic unfolding activity, and they can rip anything apart. So I don't think an open GRO-EL ring has that kind of power. I think it's directed, the whole thing is, in my view, driven by these hydrophobic contacts. So, you know, if you have a second or two, there's probably some degree of annealing in such a way that you optimize the hydrophobic contacts. And yes, that does come back to water, right? Because hydrophobic contacts are basically removal of water and entropically favorable behavior. But I think that's as much as we really know.
We could talk, I mean, later about experiments, but it's very reminiscent, the size scale of the large changes in water volumes that occur in some of the mitochondrial channels.
I see.
So there are some osmotic stress techniques that can start to access that, that information.
That's interesting.
Hi. So there are a number of HSP70 isoforms in the cytosol. And you say that HSC70 or HSPA8 or whatever it is the one that's associated. Mm-hmm. Is there anything distinct about that one that other than?
Well, I think, I mean, there seems to be a single HSC70, you know, cognate 70 in mouse, and that's the guy that's coming. The HSP70 class is, I mean, it's pretty way down the list, both with respect to binding to this misfolded thing, but also, I really went quickly through that, but in the slide where you saw some total abundance estimates, it's pretty well down there. So it's like HSC70 is the major sort of neuronal chaperone that's sitting in the cytosol. Now, is that a function of, you know, vesicular kinds of functions, or I'm not sure.
So it's an abundance? Is what you think that is? So
what's an abundance estimate?
No. I'm just saying. So the specificity is based on abundance rather than something.
Yeah. I think to some extent that's true. Mm-hmm.
Okay.
Yeah. No. I don't want to connote that it has some very specific ability to recognize misfolded things. It's the most abundant thing there. And so it has to be the front line. But on the other side of that, there's nobody else there who seems to have a higher abundance that we would have detected. You know what I'm saying? We don't see that. There's something else. Okay, well, thank you once again.
There's a reception in the library for art right after this talk.